Hey, all right, week 9, we got another test this week. So let's kind of run down everything, and then we're actually close enough to the end of the semester. I've got a few things that you can be doing to kind of wrap up and prepare for that too. So first off, though, the big one is exam 4. It's going to be open Monday and Tuesday. So that's going to be on chapters 9, 10, 11, and 12. So remember, 9 was like the cell cycle and mitosis. 10 was meiosis, so producing sex cells. Those two were really similar. 11 was on genetics and Gregor Mendel, and then 12 was on, it's called molecular biology of the gene. And that was just talking about our DNA and how it makes proteins and how it copies itself and makes RNA. So those are all, even though that's a lot of chapters, those are all pretty closely related. They're all dealing with cells and genetics and DNA. So that's going to be open Monday and Tuesday. This one is 75 questions. It's a little bit more than what you've been doing, but not a ton. And like I always say, most of you are finishing the test in 20 minutes, give or take. So don't let that scare you. You'll still have a minute per question, but most of you are not using that much time at all. Just a reminder, read the instructions for your environment video and all of the, like there's some additional instructions before you start the test. You cannot have any resources at the end of during an exam. It would be a shame to get this far at the end of the semester and then get a 0 for trying to cheat or having your phone out or notes or anything. So just make sure you understand those rules and follow those. All right, so after the test, the next chapter is 20. That's on viruses, archaea, and bacteria. So that's dealing with, first off, viruses, obviously over the last year and a half or so, 2 years, we've become really, really familiar with viruses and the disruption they can cause and the damage and everything. Let me say, though, I recorded this lecture video a few years ago before COVID. So we're going to have a section where we talk about different viruses and different outbreaks and plagues and the damage that they've caused. There's nothing in COVID about there just because I haven't re-recorded that lecture yet. I'm sure the next version of the textbook we get is going to have a ton of information. I'll update it and all that. But I guess you've heard enough about COVID on your own the last year and a half. So that's going to be like noticeably absent in the lecture video. Obviously not that it's not important. But it's just, like I said, didn't re-record the lecture video. You are going to see things, though, that you'll probably relate to that you've heard on the news, and you'll go, oh, okay, yeah, I remember hearing that. So we'll talk about RNA viruses, which COVID is an RNA virus. We'll talk about how viruses reproduce and mutate, and that'll deal with like the Delta variant that we're seeing right now. That's a mutation of another strain of the COVID virus. All right, so anyway. After the virus part, that's like the first half of that chapter. The second half is on archaea and bacteria. So that'll be a little bit different. We've talked about bacteria a little bit earlier in the semester. We're just going to get into a little bit more detail. All right. In that lecture video, we'll talk a lot about viruses and diseases that they cause, and then we'll talk about bacteria and diseases that they cause. Note cards are a really good way to take notes and study for that section in particular. So just if you're looking for a way to study, that's a really good recommendation I would say to do. All right, you've got a review quiz for chapter 20, and that's going to be due on Sunday, August 1st. Now, this one is different. One of the learning outcomes that I have to do for this class is to teach you how to identify bacteria. Now, if this was a traditional class, we would have a lab with microscopes, and I would teach you that way. But since it's not, since we're online, I had to do something a little different to kind of check that off. So your review quiz this week is going to be different. It's not going to be questions from the lecture video. It's going to be about identification. So to do that, when you go to the quizzes tab this week, you're going to see there's a document that's bacteria identification guide. And if you'll open that up, it's going to show you how to identify the different types of bacteria. So we do something called a gram stain on bacteria, and when we're done with that, they're going to be one of two colors. That helps us determine if it's gram positive or gram negative. And then we can identify them based on their shape too. So that guide is going to help you identify bacteria based on that. You can use that guide when you take the review quiz. That's perfectly okay. I actually want you to use that identification guide when you're taking the review quiz. And you're just going to name the different types of bacteria. It'll be pictures on this quiz, and you'll have to, multiple choice, but you'll have to click the correct bacteria. All right. Couple more things. So discussion board number 4 is on genetics. This one's interesting. So this is some research that I probably need to update it because there's been more advances. But In the genetics chapter, we talked about you have two sets of alleles, one from dad and one from mom. What we didn't really discuss is that there's not only DNA in your nucleus, there is also DNA in your mitochondria. And scientists have started doing this process to eliminate certain mitochondrial genetic disorders. So those are disorders that are caused by DNA in the mitochondria. They'll take an egg from a third parent, put the mother's nucleus inside that egg, and then the father's sperm. So what we end up with is kind of cool. It's kind of weird too. We've got individuals who are not inheriting disorders from their mother that were carried in the mitochondria. But what they are doing is they're having three genetic parents. Like part of your DNA is from dad, part is from mom, part is from the donor of that egg. So we're going to go through, there's a couple radio stories I want you to listen. They're both really short. But instead of like leaving a summary and responding to another student, what I've done is started two different columns. One says pros and one says cons. And you're just going to leave in the pros column something about this type of research that you think could be beneficial. You know, obviously eliminating disorders that people can be afflicted with. List a con about this type of technology or this type of procedure, something that you think could be an unintended negative consequence of this type of thing. All right. I've been accused, students like call me up saying, I can't believe like you think this is right or wrong, or like both sides. I'm not asking you to agree or disagree. I want you to see both sides of the issue is all it is. All right. So you'll leave one comment in the pro, leave one comment in the con, and that'll be what you do for this discussion board. All right. Finally, the last mandatory lab that we have is on genetics. So what I mean by that, remember, you've got a bonus lab that you can do at the end of the semester, but this is the last one that will actually count just for regular points, is on genetics. You're going to be doing some Punnett squares. You're also going to be doing some math, something called chi-square analysis. And I know this isn't a math class, and that can be kind of intimidating. So number one, I want you to just give it your best effort. I'm not going to be super picky on the math portion. Now, on things that we covered in lecture, I want you to know that. I will grade that right or wrong. So Punnett squares, ratios, things like that. But on the math portion, I know this is a little tricky. So what I've done is when you're reading through the instructions on SI Cloud, if you there's going to be little videos like this video right here embedded in the instructions. And what you'll do is you will like click the video, and it's me working through a sample problem that'll help you out with that. All right. So watch those sample problems. It'll make it a lot easier on you. Just a heads up though, it says that this lab takes an estimated 4 hours to complete. I've had students before say it didn't take that long. Like that's on the long end of how much time you should set aside. But I've had some students say they had trouble with the math and it took them those 4 hours. So just a heads up, this is not one that you can like do at the last minute and finish it and turn it in. The good news is it is your last one. So unless you want to do the optional bonus lab, and I would recommend that you do that, but that'll be the last one that you actually have to do or you lose points. Okay. If you look at the materials you need, you should have most of that already. You just need a coin to flip. That's going to be part of the lab. You'll need a camera to take some pictures. And then everything else you should have at home. All right, end of semester survey. Since we are getting close enough to the end of the semester, I've opened up a survey. It's in the quizzes tab, and it's just to get some feedback from you about how you thought this class went, the different parts, like the lecture videos. Did you think those were helpful or not? The labs, do you think those were helpful or not? The discussion boards, things like that. And it gives you an opportunity to just give me like a comment or some optional feedback. It's fine. I can take it. I'm not going to like get offended. They said, "Oh, they didn't think my lecture videos were good." That's fine. Just give me whatever feedback you want, and I'll make that one worth 5 bonus points just for you helping me out on the ways I can improve this class. Lastly, 2 reminders real quick. The deadline to withdraw from summer classes is next Monday, so August the 2nd. So all I need you to do is check your average, and if your average is not where you need it to be right now, like if you know I've got to have a certain grade for this to transfer to another college, or I'm definitely, if you're not passing right now, if you will send me a message and give me like a minute and I can figure out what is it going to take for you to finish with a passing grade or to finish with whatever letter grade you have to have in this class. I can just pull up the grade book and calculate that real quick for you. The other thing is the makeup exam is next Monday and Tuesday. So that makeup exam is going to be open the 2nd and the 3rd. Not many people have missed any exams in this class, so that's good. If you haven't missed a test, you don't have to do anything with that. Just ignore it. But if you miss an exam, remember only one exam can be made up per semester. But if you'll take the makeup exam, I will grade that, and that'll take the place of the 0 for the exam that you missed. Now, if you've missed 2 or 3, you can only make up 1. The other missed exam will still stay a 0. But it's, like I said, it's better than a 0 for at least one of the missed exams that you've had. All right. Wow, that was a long video. I'm sorry. I didn't realize I was rambling that much. So if any of you are actually still watching at this point, thank you. And if you have any questions, please let me know. Thanks.